All right, everybody, I'll take you through some of the things I've been thinking about while I've been here. So I'm not going to tell you anything about my research. I'll do that on Tuesday. And this is work that I've been doing while I've been here, mostly for my own edification, but hopefully there's some interesting things for you guys. And the argument that I'm going to use is summarized here. It's complicated, and I'm going to basically present some natural history and some explanations to put together what I think might be a way that quantum mechanics is interacting with biology. And this is highly speculative at this point, so keep that in mind. And I'll, of course, ask questions as we go. So the argument is going to go like this, that quantum superpositions are being used to find unlikely solutions to a whole bunch of problems in biology. And that to do this, biology has actually created these things I'm going to call BICs, which are biological isolators of quantum superpositions. And that this gives a biological entity the ability to deal with many, basically the unlimited number of problems that are faced by life. And my starting point, as always, is why are there so many viruses? And as you probably know, or I hope you know, is that viruses are the winners in the game of life. There are a lot more of them, and they use a lot of the energy and resources on the planet, and they're by far the most diverse things out there. So they, by almost any definition you want, are the ones that are winning. The other, and the reason I went to quantum mechanics is really because of this, that viruses are actually really small. And this is, I guess, intuitive to a biologist, but they actually seem big to a physicist. So we'll go through why I decided to put these things together. So there's a thing called Planck's mass, which is, depending on who you ask, the approximate cutoff between the quantum and classical worlds, the physical worlds, somewhere in that range. Though I think most physicists would say Planck's mass is quite big for quantum behavior. And to give you an idea, it's somewhere in the microgram range. And viruses are quite a bit smaller than Planck's mass.
It's not hard to calculate, because usually the cutoff is h, so it comes at the quantum of effect, which is mass times velocity. So probably it depends then on which velocity you...
Yes, exactly. So it does, this is, this depends on rest mass in this case, right? So, yeah. Just to give you a feel, this is, it depends on your plane of how your relative movement, right? So, all right, so this gives you a feel of about where viruses sit, or at least where phages sit in this game. And the other, basically, very classical way of thinking about quantum behaviors is to actually start looking at the two-slit experiment, which I'll go through just a little bit later on. But to give you a feel that people have successfully performed a two-slit experiment with biological, organic molecules that are in this range, about 3 orders of magnitude smaller than a small virus, roughly somewhere in that range. So for the biologists, there are going to be 2 rules that I'm just going to lay out there, and we're just going to have to accept that they're true. Okay? The first is the wave-particle duality. And this is, of course, one of the real hard things that people find confusing about quantum. But the idea is that everything is a wave. Okay? And when it's measured at that particular instance, it becomes a particle. The way that I'm going to use it is, it's hard to talk about it without math. So just imagine that as the wave is moving, there will be a place where it gets measured, and when it gets measured, all of the energy and all of the mass associated with that appear in that one point. This gives you some very weird behaviors, which are the ones that we don't like from a classical point of view. So this is what it would look like in a two-slit experiment. So you have the wave form of an entity coming in. And then if it goes through the two slits, you start getting these interference patterns. And this is what you detect on like film or some other material, depending on what you're trying to look at. So some detector. And each time one of these waves hits here, it becomes this particle. And all of the energy associated with that wave is right there at that point. And then, of course, if you do a lot of them, you get the interference patterns. Okay? The wavelength is determined by de Broglie's formula here. Here is the wavelengths for a number of things. So here's a buckyball. So these are a little smaller than things that have been used in classical to slit experiments. Here are the phages, and here's Planck's mass up here, calculated for the relative velocity of the system we're looking at. And then these are all the cells, right? So notice that essentially all of life exists below Planck's mass. Now, this is just natural history to get you, I think this is important to look at. So what we see, we normally think of quantum effects as really being only on photons or electrons at that level. So this is an experiment where you use a scanning tunnel microscope to arrange iron atoms on this copper surface. So they've taken and they move them around, okay? And then you move across with the microscope and you look to see, you're basically essentially bouncing off the electrons. You can think of it that way. And what happens is when you create this electron corral, you get these standing waves of the electrons. This is your lowest energy one here. And then you get less likely energy versions going out this direction. And what's important for my argument is that these things are actually fairly large. They're in the hundreds of angstroms range. And you can see the quantum behavior at that point. This is where you just take that same thing and you impose it on a phage head. So here's the phage, this is where its DNA would be, and this is its tail. These are what are floating out there. About 10 to 31 of them on the planet. And they appear to me to be right at that perfect level of where they might be able to observe quantum and classical behaviors. And that's going to be my argument.
Just to be clear though, on the previous slide, it's not the iron atoms that are behaving.
No.
It's the electrons.
It's the electrons, yeah. So right, let me make sure that that's clear. This is what you're seeing, the electrons. And if you want to pick that electron, if you want to grab it, and it's interesting from a biologist's point of view, you actually want to be right here. And if you're a little bit off, couple angstroms off, you're going to actually be in a very unlikely place for that electron to be. Further on, you'll have another chance of grabbing an electron, et cetera, right? So you can actually see this. This isn't, of course, how it would look in a biological system, but it's not so different. All right, the second rule that I'm going to deal with or use is that there are these things, the superpositions, right? So when you're spread out through that wave, you have, the electron is actually in all of the places at the same time. Right? And this is, and it'll stay in all of those positions effectively until, and when it's in that form where it's going to work in like the two-slit experiment, it's coherent. When it interacts with something, it becomes decoherent, and that's when it collapses into the electron. And the process of measuring the quantum waves and making them into basically particles where you know where they are is this, and making them decoherent is E-selection, I can't say it, or in-selection, something of that nature. And this is a proposal about how you move from the quantum world to the classical world. There's many other ways to think about it, but that's the one I tend to like the most. So what does this actually look like? And here's a more solid example from the chemical point of view. I'm going to use P450 cytochrome, which is an important, a very important structure in biology. What it is it's a ring of carbons that coordinates an iron-sulfur group to attack double bonds, double carbon bonds. And we know that P450 is, if you model it in the classical sense, and this is your kind of your classical version of it, it actually doesn't measure, you know, where you have s orbitals and p orbitals and so forth. It actually doesn't match what we measure in the lab. We know it doesn't work that way. Okay? In fact, the only way to match what we see in the lab is to use one of these quantum mechanical approaches. And that's very hard to do with one of these really large complexes, but people have done it because this is so important for detoxifying and in particular drug inactivation. And the way to look at this graph, because I'm going to use this later on, is that these are your energy barriers. Okay? So this is your starting point of the reaction, and you want to get to either side here. And these are the energy barriers you have to go over to get there. Okay? And classically, how we think about it is that you have to have enough energy to get over and make it down. That isn't really how it works. And this is the, I'm just superimposing. This isn't really how it works, but just remember, here's my lowest energy state for my electron. And then I have different ones coming out, so I'd be here. Okay? And then if you imagine, as it spreads out into less and less likely energy states, you actually can go over these energy barriers and end up in one of these wells over here. Okay? And that's what electron tunneling is. Okay? Good, everybody? And that's caused essentially because of uncertainty. That's one way to think of it. All right, so the proposal is that life would use these sorts of behaviors, and why would it actually do that? And this, I think you can understand as a biologist, that every organism, every virus, every cell out there has a limited resources available to it. And these superposition searches would allow you to actually look through a whole bunch of different possibilities with many fewer mechanisms. The other thing is that superposition searches are extremely fast. So they're measured in the atto to femtosecond levels. And most biological reactions are going to be many orders of magnitude slower. And they allow you to go through these energy barriers that would be forbidden through classical behavior. Now, the reason that most people would say there are no quantum, or there are no real large-scale quantum behaviors in biology is because it's large, which means that's where you get measured. Remember, so if you get measured, you go decoherent. So being large is the best way to get measured. It's warm, right? So basically things are vibrating and running into each other. And wet really matters a lot because that measurement could just be a water molecule running into you. So this leads to decoherence. To get around that, what I'm proposing, or what my working hypothesis is that there are these ways of isolating to avoid decoherence. And biology or living organisms are doing this by keeping things small and using the natural equivalents of electromagnetic fields and light traps. And I'm going to focus mostly in this region here. All right, so why is it at all possible? Well, here's an example. We are already doing it. So if humans can do it, my argument would be biology has probably already figured that out. This is an example of something that's actually quite large. It is, this is a harmonic oscillator that was built at UC Santa Barbara. And it actually, you can cause it to vibrate, and it will be in a superposition that you can measure. And these things are large. They're 700 times the size of a phage, okay, when it's doing that. Okay. This is another example. These are used all over. You guys know them as quantum dots. This is a double quantum dot, which is used, that's what the quantum computer people want to use, or they think about a lot. And this is effectively a qubit sitting in a solid state structure. Again, it's large. It's much bigger than much of the things we think of as biology. The other thing is we have examples from biology. So here is a thing called the carboxysome, and these belong to these microcompartments, MCPs, that are found in bacteria. And in some bacteria that carry out photosynthesis, what you do is you organize your rubiscos inside these carboxysomes, and the idea is that it's probably a CO2 concentrating mechanism before you cause things to go. And this is, we know that actually there's a lot of quantum behavior in here where there's entanglement and and teleportation going on within this structure here. And I just want you to see that here again is that quantum, or that electron corral. It's still in the size range where we would expect there to be a fair number of quantum behaviors. The other one, and the one that I'm going to focus on, is a little bit of membrane vesicles. And I'll show you why. I just want you to remember that membrane vesicles are extremely complicated. So there's literally thousands of them, and they're one of the main things that in the evolution of life that really led to different things. In particular, development of the eukaryotic cell, though a lot of bacteria also have membrane, smaller versions of itself. And what's important about this is this here. So the way that you build a membrane is with these with something like a phospholipid, where you have the hydrophobic head and then the hydrophilic heads and the hydrophobic tails. And what actually holds this together is pushing them together by the actions of the water. They're not actually explicitly attracted to each other in this direction. And this creates this zone, which I'm hypothesizing is an important place where you can get quantum isolation, which is right at the between the two leaflets of the phospholipids. Just remember that in the leaflets, what happens is that you can migrate within your leaflet really easy, but you can't flip from one side to the other very easy. It takes a lot of energy to do that. And the proposal is that this is causing, it's right here, it is our quantum isolator. The other thing that's interesting about membranes, of course, is that they actually have very strong EM fields working across them. So here is, it depends on your organism and what environment it is, but you get charge distribution across it. And the membranes themselves are extremely good insulators. So they're very good at keeping electrons from moving back and forth. And you actually end up with really fairly strong potentials across them. Quite so much so that this would be many orders of magnitude higher than what you would use in a lot of the electronics that we would be interested in doing quantum work with. And finally, there are other things in biology that suggest that we might have magnetic EM sort of isolators. And these are things like the magnetosomes, which are found in bacteria. They're also found in like bird ears and so forth. And these are very strong magnets. They're basically as strong as you can make a magnet. And we know that they create EM fields within some bacteria, and that they organize the enzymes. So exactly what that does is still it's still not known. So what would one of these things look like? And this is my proposal. So I'm going to call them BICs. And they're going to be lipid-based for this thing. And they're going to look something like this. So you have the membrane with the outer and the inner leaflets. Okay. This is important. You have monotopic proteins. What that means is that they're only on one side of the leaflet. Right? So they can only be in this leaflet or that leaflet, which there's a lot of examples. And then I'm going to be using the P50 system P450, because it's also monotopic. It's only represented in one of the leaflets, as is its reductase. And then I'm going to use a toxin because that's what the target of these guys. And we know a fair amount of how these might work. There's more of a physical look at this based on some recent work here, but also there were classical studies in how you might have a quantum, quasi-isolated system. And where you might want one is exactly where we're finding P450. It's where you have to detoxify, in this case, a whole bunch of different varieties of stuff. So this is just a small sampling of what you actually have to deal with when you eat stuff and when you're living in the environment. There's essentially an infinite number of these really complicated ring structures out there. So how this would work is that your toxin would bind to the protein and be imported inside of the BIC. Okay? And so at this point, you're isolated. So you've only got the toxin. Nothing, this is our quantum isolator field here, at least according to my world. And then what happens is that you can interact with the P450. And remember, at this point, if this is really a quantum isolator, this thing will be in all of those superpositions, as will the whole system actually should be in a quantum superposition, which will allow it to get over those energy barriers that I was showing you earlier. This just gives you an example of why we would care about this. So we know that there's thousands of xenobiotics. We also know that P450, the protein that holds that cytochrome, are extremely diverse. There's hundreds of them out there. Well, actually thousands of them. Humans have about 57 of them. Mice can have 100, but you find them in bacteria and viruses, and everything seems to carry them. Okay. They're monotopic, as is their reductase, so they only sit in one leaflet. Okay. And we know that they seem to work through a tautomeric mechanism. So they do fit all the criteria. And that's just to remind you of what that looks like. So they can tunnel from this barrier to that barrier if you give them a chance. Okay. So the idea that would be you go through that quantum superposition search, and then if you get the right reaction, which is one of those things that happens when you're in these superpositions, and you grab it, you actually fix the whole system into that state. Okay. And then you flip it out. So that looks actually very similar to a whole class of biological reactions out there, which don't really require any energy, which sounds really weird, but that's what we call them, is energy-free biological reactions. The energy has to come from somewhere else at some point in the system, obviously, because you get some sort of friction, but there's a lot of things along this line. So this is my proposal of how it would look like. And we've actually got a couple papers that look at this from a classical thermodynamic mechanism, which I can show you if anybody cares. Yes?
I get what you say, but this
Yeah.
can also be explained by just the classical
It can't.
enzyme.
No, it can't. So that's the problem. It cannot. This is the explanation that works, that matches the classical. The classical mechanism doesn't work for explaining.
So here's my version. I would like to hear your opinion.
Okay.
So when it's acting like an enzyme, you grab your substrate so close to each other.
Yeah.
And the result, it decreases the activation energy.
Mm-hmm. Yes, that's the classical explanation.
Uh-huh.
It's just that we know that isn't actually how it works. That there's everybody has this idea that there's cavities, right, and things can vibrate in there, and that's, for most things, that's close enough to the truth.
Okay.
But in actuality, it looks like you have to use quantum explanations to really see it happening, how it's working.
But you said you didn't explain why.
Yeah, so I don't know. It's not, they don't vibrate in the same way that they can grab, or the electron can be coordinated exactly perfectly. I'll show you, I've got
Okay.
something that looks a
Okay,
little
the,
like
the
that.
second point is about
Yeah.
similarity. Even though quantum can work, but still cannot against the classical similarity. Which means, there's no free energy.
There's no free energy. No, that's not the point. We
Okay.
know, but we also know that there's there's a whole large class of proteins that or reactions that people would say are energy free in quote unquote in biology, in the sense that we don't know what the energy source is that's driving the system.
I think what I take is, you don't need investment in energy. It's, their overall reaction should be similar and favorable. Nothing can go against the classical similarities.
That's yes. I don't think anybody would argue that
Okay.
that's going on at all. I would just say that there's definitely a big disconnect between that you have to have some sort of energy in any reaction and the observation that you don't need any energy. The classical one, or the one that I think people, or that I get hung up on mostly, is like a recombination event. So recombination events can go back and forth. Right, you can do an attack, a covalent attack with like topoisomerase. You can put that thing in the freezer. You can let it sit there for years on end, bring it back out, and cause it to go backwards. Right, so this is considered energy free because you don't have. There's nothing that's clearly identifiable as energy in the system. It doesn't mean that you don't have energy going in. Yeah.
I mean, passive diffusion is also energy-free in a sense.
Yeah, exactly. There's
a lot of carriers and canal proteins which don't use energy. It's just the concentration gradient which drives the export. So for me, it's not
you can
or not a problem actually.
It's not a problem. So I'm not saying that this is a problem. I can come up with classical explanations of everything on the planet right at this moment for this stuff. What I'm saying is it's a lot faster. Remember, it is it's like 4 or 5 orders of magnitude faster depending on where you are, right? Which does matter in biology.
Yeah.
Yes?
Can I just be clear? So what's
Yeah.
the quantum bit here? So the reaction of the P450 is using quantum mechanics.
Right.
That's fine, but is there more quantum mechanics?
No, I'm just saying that this is giving it the ability to do that. And there are examples in the literature already where there seem to be behaviors like within the bacterial chlorophyll systems. There are places where we seem to see sort of enough isolation of the quantum world that you can get quantum behaviors. So this is, can we go
so it's
bigger?
the isolation that's important.
It's just the isolation because
Right,
it
so
lets
it's
you
not just
play.
that the enzyme is behaving quantum mechanically, because that's fine, that's well established.
They all behave this
It's
way.
because it's inside the vesicle, and that's preventing it from becoming decoherent.
Yeah, it's just slowing down the time to decoherence. And you only need to gain a couple nanoseconds actually to gain a lot in these games. Yes.
You call it large scale. I would call it large scale quantum coherence, right? Because
Yeah.
all chemistry is quantum mechanics.
Exactly, yeah,
And
yeah.
so it becomes confusing.
It does actually get very confusing, and I'm still struggling exactly what I'm trying to say. I have to give you, but yes, it would be large scale. We know of a large number, or sorry, we know of a number of large scale quantum behaviors, so it's not completely outside the realm. It's just that it's very hard, for the most part, to convince anybody to look at larger scale quantum behaviors in biology. And I'll show you just a little bit more, and because I don't want to torture you too much with it. Okay? Here is going a little bit to what Chu was asking. So the problem is when you have something like so you have the P450 protein, and it's holding the cytochrome in there. All of these things are built to hold a particular substrate in there that it can attack. So you've got the coordination of the electron onto the double bonds. And what that means is that to solve all of all of the possible things that you could put into that pocket, you would need like thousands and thousands of pockets, right? Or you would need a cavity that vibrates a lot, which is the classical explanation. What I'm saying is that by taking advantage of these quantum superposition searches, you actually are using this, you're actually holding it even when it's not quite in the wrong orientation, and you're taking advantage of unlikely possibilities by giving it time to look through these superpositions before it goes decoherent again. It's just like how a quantum computer would be running in many ways. And I think there's tests of it. So this is an example of how you might be able to get a molecular test. So you take and you build it. This is a molecular biologist looking at this. So you basically, in a test tube, you're going to put lipids to make the vesicles. You've got your monotopic transporters. You've got the P450 cytochrome in its protein. And you want to limit this. This becomes important in the actual experiment. So that the stoichiometry, you're going to have about one of these per liposome, approximately. And then you have the reductases so that you have a lot of them so that they can recharge this cytochrome. A buffer and an NAD pH. You sonicate to produce the liposomes. Sorry, that should be an O. What happens is that you end up with, of course, some little vesicles that are, they just have the buffer and the NAD inside them. Then you're going to end up with other ones that have the P450 on the inside and hopefully, and reductases. You'll have other ones that have them on the outside, and then you're going to have ones with both of them. To start the reaction, then all you do is you add an optical sensor for P450 activity, which there's a whole variety of these. And your expectations roughly are that you're going to get a total activity where this is chewing up those double bonds. Okay? And you expect more in the liposomes than you do in the bulk. Okay? You're starting with the same concentrations of everything, so there's going to be a couple controls that you need here, but you should actually be able to directly test it. And then you can actually go back with an antibody to the protein and see, was it on the inside or on the outside? And you would expect this one to have the most activity. Make sense to everybody? So this is a test, and it's a reasonable test that you could actually do. The other expectation is that when this is going to be best is when you're using the P450s from different, that are not very good at whatever substrate you're throwing at them. So we know a lot about this because drug companies care about them. So there's literally hundreds of different optical versions of this that you could use. There's thousands of these depending that you can make. And so you start mixing and matching them. So you measure what they do in vitro, and then you measure what they do in a lipid system. So that would be a molecular test of this sort of thing. There's also physical tests. And I think actually this one may be the best one right here. So we've got now the different optical techniques that tell you about whether you've got quantum behavior going on. Well, that's, and in some of these would probably work within cells. So you can actually look for the vesicles in the cell. One of them that I ran across here was a way that uses quantum entanglement to look to see whether or not you have quantum behavior. Okay? And these actually work with fluorophores that we normally use in cell biology. So I'll bet you that the data is already there. Okay? And we already know that this is true for some things out there. Okay? The other thing is that you could do, so these are a little bit more science fiction like, but you could look directly for tunneling of something large like a phage across, for example, a gold leaf. Because you can make a gold leaf so it's only a 1 atom thick, and you could actually see, do you ever see where a phage jumps across that interface. And then the even harder one to envision, but would be classical, or would really, I think, convince a lot more physicists, is could you come up with a way of making a two-slit experiment where you're just using the inner sphere of the lymphosome? So this piece right here, could you actually use it in a two-slit experiment? And I can kind of envision how you might do that, but I don't think that there's anything practical that you couldn't actually do it right at this moment. I don't know how you would maintain this in a field where it's moving. All right, so those are some tests of it.
Hey, talking
Yeah.
about test, can you actually put in something between the two lipid layers that would kill the isolation?
Yeah,
process?
and I should have mentioned that, yes. So anything that goes across the leaflets would, in theory, connect it to the environment,
yeah.
and that would cause, so you can do that in that experiment. I was supposed to say that, good question. So you should be able to kill it by breaking this quantum isolation effectively, would be the idea. So there's a lot of controls that
But
could,
that should be easy, no? I mean,
Yeah.
in a way-
Proteins, for example.
What?
Yeah, proteins. Usually there are a lot of
most proteins will do it, yeah.
But
No,
then,
yeah.
your test would be whether inserting a protein in a vesicle would change the kinetics of your detoxification.
Exactly.
That's a pretty simple one. No?
It's easy, yes. It's not so easy to do that experiment.
I mean,
It's not horrible, but it's
Yeah.
going to take someone a couple months to do it. Yeah. This one, I bet you that the data already exists. That's the thing. I just don't know where to go looking for it. I'm not exactly, because it would have been using a confocal microscope where they were measuring decoherence at some point. But I don't know of a good example. Some optics person might know that for you. Right, so then I'm going to take you just a little bit further and talk to you about this part right here. And this is, there's this observation that you have all of these P450 proteins, okay, spread throughout all the domains of life. So how did you end up with all of this diversity? And to do that, I'm going to break my thinking into two parts. Something where quantum ecology is going to be using these BICs to detoxify, like we talked about, build things, etc. Okay, so do unlikely sorts of things. And then quantum evolution in the sense of not like, this is quantum evolution, not in the sense of big jumps, but actually evolution that uses quantum mechanics to do its job. And I'll show you what that means in just a second. But basically, this is a remembrance of this process right here. Okay? So the hypothesis is that there's a feedback between the ecological solutions and the evolutionary mechanisms. And this goes to something that a lot of you probably know tons about, and I know more about than I want because of the people I hang out with. But this is the Karen's experiment. Okay? And this is a famous experiment where basically they went looking for mutations exactly in the spot that a cell needed to survive, right? So instead of having random mutations and selecting against, it was actually, can you get them in a particular area? And what they found and what they published is that there was actually this non-random distribution of mutations, and that was called adaptive mutability. It turns out that there are extremely nice classical explanations, so just Darwinian sort of mechanisms based on a number of different things, but expansion is my favorite one, by Roth and Anderson, which shows how you got this sort of game. Okay? But it also led a number of people to really think about other mechanisms of evolution. And that's what I want to talk to you about here. These are broadly quantum models of adaptive mutations. Okay? And these came out based on this other experiment. And McFadden, who's from Surrey, and O'Gratz, oh, I can't say this. He was, I think, in the States. He's now in Paris, and these guys are in Oregon. They actually basically all came to the same sort of model, which is that these tautomeric, so just like we have with anything where the hydrogens are moving around or the electrons are moving around, you get these tautomeric versions of the base pairs. And this was originally actually suggested by Crick when he was still here in Cambridge, that this could be a way of doing evolution. And then what happens is that because of how the hydrogens, let's think of it as hydrogens, move around on the bases, that you could actually get non-standard base pairing. All right, and this is what one of these schemes looks like. This is McFadden's version. It doesn't, it's a little, sorry, I drew it and then took a picture of it. But the idea is that you have your DNA, and if it's in a superposition, you get a tautomeric base, for example, here, okay, which then gets translated into a different amino acid. Okay, and then that amino acid is queried by the substrate. Okay, so imagine that this was an active site, and this active site could interact with the substrate. And this measurement event here propagates itself back and fixes the tautomer in the original DNA sequence. Okay? And it does this thing through, it's called the reverse Zeno quantum effect. That's the argument. Okay? If you go this route and it can't interact with the substrate, you don't fix that tautomer, right? So you don't get the mutation. Essentially, all of these things look like this. Good? Does that make sense to people? So this is a true quantum behavior. So you're making your mutation exactly where you need it based on your interaction with the substrate.
How does the communication go backwards?
Yeah, so magic, but it's called, so the idea is that the measurement event itself, it locks it in this, because you don't, because of the uncertainty principle, you lose where you are in the superposition, right? And then by making the measurement, this is actually the superposition you select for. And there's a whole bunch of different ways that they explain it, but that's the idea. Okay? And
Yeah, at the beginning, it's not anything, and then the measurement fixes it to that.
Fixes it to that, yeah. That's exactly it. And in fact, there's a lot of, as you can imagine, problems with this. Okay, so the big problems is that it takes way too long. There's no way you would avoid decoherence in the system, because you actually have to go through transcription, translation, and you have to do that quantum feedback part. So you have to do the measurement, but you get this is like, milliseconds. It's not even in the nanosecond sort of range. Also, it's gigantic, right? So you can imagine that the mechanism here, basically you have the chromosome, you have an RNA polymerase, you have an mRNA, ribosomes, and the feedback, the quantum feedback system going. Again, a whole bunch of stuff happening. So really, it's almost impossible to imagine how this happens. So,
Can I ask?
yeah.
When this is proposed, you say almost independently by three groups. Right?
Yeah.
So they presumably knew about these problems or pointed them out. Didn't seem to.
Right.
But I still thought it worthwhile to.
Well, so they went into three basically different versions of it. Okay, so the Davies group, I'm going to probably mess this up, but the McFadden group invoked the quantum Zeno effect, but they did it as a reverse quantum Zeno effect. So basically, the measurements as you go back through that chain is what's locking it in. That's their mechanism.
Oh, okay. Because they're physicists, those people.
Right, so they know, they can at least, yeah. He actually invokes the idea that the cell itself is essentially in a superposition. and he has a paper, a follow-up paper that came out like just last year maybe, that suggests that he draws, the the eigenvector for the cell, blah blah blah.
This
Yeah,
is very big, right, this cell, so I don't see how to.
I don't believe it either. I'm just saying that that's what they do, and I forget actually how these guys get around it. To tell you the truth, I'm blanking on that. But they all use this essentially same idea. My proposal would be, if there's something like this going on, you can actually make it a lot smaller and you can put it inside one of these big things I'm talking about. So what you have to do is you have to make it small and quick. Okay? And so what I would propose is something like a translation-free query. And how this would work is that your DNA would make your mRNA, and then the mRNA would be where because it also has tautomeric bases, so you'd have an mRNA enclosed inside one of these bits, and then it would go through these superposition searches with just the tRNAs there, and with no translation, right? So we know that this can work in some systems, so that where the mRNA acts as the template to hold the amino acids in the right place. You don't form the peptide bond here, so these are loaded tRNAs that are going on and off. And then you capture one with your substrate. So it's the same exact device that I was telling you before. And that fixes your mRNA. This system breaks down. You do a reverse transcription, homologous recombination, and you fix it that way. Okay? So all you've got in this system is an mRNA, loaded tRNAs, and your substrate to get there. This isn't completely crazy. There's a set of enzymes that actually do exactly this. In fact, there's a number of different versions of them. And there are ones that even seem to be almost specialized to do some parts of this, which the CRISPR-Cas system is a good example of where you might be able to just use that. You can also imagine that these things are broken into even smaller pieces because you don't need the whole mRNA to do this. So where would you expect it to happen? Oh, right. So this is just a calculation. Remember, these are these wavelengths. This is the buckyball, remember, which we could, we know does 2-slit experiment stuff in this area. This is if you break it down with that modified version that I just showed you. So it sits somewhere here. And then this is the original size, according to those other models. So if you make it smaller, you're definitely closer to where it might work. Here's something, again, natural history. These are called stress granules. And what happens if you stress out a eukaryotic cell, they actually end up building these little inclusions here, which are mRNAs that are caused by basically causing the polysomes to fall apart. Okay? They're small and they're ribosome free. They have tRNAs associated with them. They have everything that you want except they're not membrane bound. So you have to have some other way of quantum isolating them than what I proposed with the lipids. There's also nuclear versions of these, which I can't tell whether they're membrane or bound or not. And we know, of course, that bacteria do similar things or many bacteria do these when they're under stress. Okay? So possibly you could have things that aren't just evolution in the sense of from one organism to the other. You could even imagine that there would be somatic versions of this that imagine in your liver that instead of having things like B cells or T cell arrangements, that you actually set up a detoxification system that's specific to a certain group of toxins through some sort of mechanism. All right, so I've tortured you guys with a lot of these things, but these are the reasons why I think it might matter. First of all, in the ecology point of view, instead of just having tons of biological diversity, you actually have very smart diversity, is what I'm going to call it. And this is, we know we use things like this all the time, where we have immunity things where we actually have the same thing doing many, many different jobs. That's what we do with the phages and the BAM immunity. So you could imagine that a system that helps detoxify or a whole bunch of other processes that allows you to deal with a whole bunch of diversity with the same system would be extremely valuable. Okay? And places that I would look for are communication things. Smell and synapses are good places to look. If there are things like the quantum evolution via these mechanisms, that would very strongly change how we think about evolution. It's basically a directed evolution mechanism, which would be really exciting. And there are good places to look for these things. I think the antisense world, the CRISPR-Cas, and a whole bunch of other things built to do that are good places to be looking at. And you could imagine these somatic mutations being important at the tissue level. And then finally, it could inform backwards to quantum computing and to physics. Because if life is building things like this, these would be effectively what we want in a quantum computer. And we could hopefully figure out how to actually use them. This is, the last point is that you get in here at the heart of what, there's a famous physicist that called the heart of quantum rotten, and the reason is this problem, this measurement problem. So how you decide to make a measurement and the interactions between the classical and the quantum world is still a big unknown, and it's important for all of these steps as well as our general understanding. All right, and then I'll just leave you with this one last idea. A perfect place and things that look exactly like what the big things I'm talking about are the synaptic complex. So here's a synaptic complex. These are all the vesicles going back and forth between them. And these things are all tautomeric. So you could imagine that how you go back and forth across the synaptic complex could be diverse, much more diverse than we think about with the neurotransmitters and so forth. This is just to show you maybe there's money in it. There was a patent. And then finally, I'll just thank the people that helped Anke, who's had to listen to this for the last 2 months. Sandy, who's a PhD student in my lab, who's done some of the calculations for me. And then, of course, the INI for letting me be here. And then I'll take any questions. I hope, yeah, we have time. Did that make sense?
Are there any other systems in place except of double lipid membranes?
I still think that there's something about the electromagnetic fields of biology that are important for this, but I could not come up with a good test or a good mechanism at the moment. I do think that it's very suspicious that essentially most of biology is really about moving electrons from place to place, right? And they do create really strong electromagnetic fields, which when you are we already know they use light guides, right? Which is the one thing we do. And if we were going to isolate things, we would use light or we would use an EM field. And that's how we quantumly isolate things. So it seems like it is a good place to look, but I haven't come up with any great ideas how that would work.
So are you actually building some of these bits?
I will have them try it. Though before I do that, I'm going to go and talk to a couple physicists and just ask them, are there any measurements that have already been done with microscopy that would give us an idea? Because remember, the idea is inside one of those vesicles, for example, you should see or longer quantum effects, basically. So you should get longer entanglement or something of that behavior. So that would be the first step, I think. But to prove it molecularly is, of course, the thing you'd really have to do.
Because there are two separate groups in Glasgow who do build those sorts of things all the time.
Exactly, yeah.
So it'd be very relatively easy to do.
Yeah, I don't think it's totally, well, I know it's not really that hard to do. It's just
I mean, and
doing it.
I mean, they're very good at actually manipulating the vesicles, so they could probably design something such that the P45 was always on the inside or such.
Cool. So I should get the names from you because,
Yeah, if you're interested, I mean, you could even go up to Glasgow. I mean,
Yeah.
and they're not biologists, so they would probably do this stuff.
Right, exactly. That's always
But they're engineers.
Yeah. We're pretty bad. Yes.
I mean, I think biology must have an edge in terms. I mean, the last point you made. So I think the known examples, they're not. I don't know how much they're believed, but it's the photosynthetic apparatus of green sulfur bacteria, navigation in birds are supposed to be quantum, but the interesting thing about it is it happens at room temperature.
Yes.
Whilst all the other quantum computing, you have to go and cool the stuff down to sort of tiny degrees. And
Right.
the dream is to do it at room temperature, but that's not happening. So biology, and there is actually, I've got some people that maybe you would like to talk to, but there's somebody over in physics who simulates these complex photosynthetic complexes. And the key seems to be how biology manages to engineer the structures so that they preserve these mysterious quantum coherences. So I'm not kind of skeptical about this, but it'd be interesting to know how prevalent is it. How many of your kinetics are wrong or evolutionary dynamics? Because is it subtle? How does it correct on what we know already?
Right.
Is it
So my guess is that it's hidden from us because of how we do our measurements. So we, for the most part, would just assume it's all classical, and then we would measure like the Km and the Kd of an enzyme and just say that's how it's working. Whereas underlying it, there's probably, there's got to be all these quantum effects, as we know, going on. What I'm looking for are ones that give them real advantages under certain circumstances. But
you should see sort of integrated advantage, right? The organism should just perform that much better. I mean, maybe it's our modeling that's insufficient as well. Maybe our ability to actually predict what microorganisms or biology in general does is really quite poor, and so we can't even compare the quantum and
that's kind of exactly how I feel is that, and the problem with our current approach in biology is that we're not getting mechanistically to how it works. We're just describing it for the most part, even at those levels. And that's so you can tell, like when you talk to the biologists, there's always it gets fuzzy. Well, it gets fuzzy for everybody, right? When you really think about how an electron is sitting somewhere or a hydrogen atom is sitting somewhere. But I don't think that we should ignore the quantum mechanics, the possibility that the quantum mechanics is really playing an important part in it. Yeah, that's my argument at the point. Alan, did you have a question? Yeah.
No, I don't have, but I mean, I think probably there are people who can probably estimate what the coherence time of these structures is.
Mm-hmm. Really?
I guess so.
Yeah,
Yeah.
I don't know who has. I mean, I'm still looking like desperately.
There's this guy called Nicholas Hine who's in physics. He's a fellow, and he does simulations of these complexes. So actually estimating it can be done, but it's it's not. I don't know how good an estimate you get just by doing it. But they can actually simulate these structures and see.
When you say these structures, you mean?
I don't remember the details. It's some part of the green sulfur bacteria photosynthetic apparatus.
Right, that's probably the best studied one where it makes that cool ring structure, and they have tunneling between two parts of the complex. No, that's what it. Yeah, I'm still working on where I would even look, to tell you the truth. But any idea?
I think you probably have to start to talk to people who sort of interested
Yeah.
in, say, quantum computing or that kind of thing, because they may be able to then direct you with other people or something like this.
Well,
Yeah.
I was talking to this guy about B12 because Simosis is predicated on B12, and he got very interested. He said
Yeah.
maybe that's got something funny going on
Yeah.
about it as well.
So that would be the perfect place, actually, because in how do you say enolamine, degradation by B12 occurs in one of those carboxysome things. That's actually that in salmonella. It's really important, actually. And there, the way that they get in and out of the structure looks like it works like an airlock. It's really cool. So they grab a volatile on this side, collapse it, and then it comes on this side. So that would give you an isolation possibly, right? Do you know that
I
system?
don't know it at all.
Yeah, I'll show you the
I'm
paper.
chemically very
Yeah.
ignorant.
It's really cool, actually. So it's not a bad place to be looking for this sort of stuff. Yeah, B12 is cool always. Yeah.
So maybe I don't get it at all, or maybe I missed it. How does this link to viral diversity?
Yeah, so I never got there. That's where I started, and I just never got there. So I'm still.
But what do you have even the vaguest of?
There's something about, yeah. So my biggest thing is there's something about those the dipole moment of the virus that's really important in something like this, but I have no idea. They encode things like this, stuff like that. The CRISPR system is an antiviral system in some ways, but it's also a way of, they also carry them. So when they're invading, there's a whole bunch of stuff like this is going on.
Is CRISPR actually sitting on membranes on these zygotes somewhere? Or?
It doesn't have to,
It
no.
doesn't, okay.
No. I just think that mechanism is interesting, right? I should quit though, honestly, because we still have another speaker, yeah.